Actually, HPV is the human papilloma virus, which are very much related to the female reproductive tract, female reproductive tract, mind reproductive tract. It is very much associated with or related with female reproductive tract. Show you a video. From this virus, there are several HPV, that is human papilloma virus, but some of them very much harmful. When it is entered into the female reproductive tract, if it is not identified, then after some time, it will make cervical cancer. Am I clear? So, you must have to identify when these human papilloma virus or HPV enter into the female reproductive tract. If you take necessary steps, then, or if you take the vaccination or anything else, then it will not move towards cervical. This disease is purely preventative. And it's not only in my country, India, not in Africa, but also in Nigeria. I will show you what till death. How the female peoples are being affected by these missing specialists. I am a mathematician. Earlier, even my previous talks, I have told you that our target is in the of biological sequences, whether we are able to formulate any kind of mathematical model. Okay. That you are able to formulate a mathematical model. But what will happen? After formulating the mathematical model and its in analytical analysis, along with numerical simulation, we try to give some, considering those who are practicing or cases today as a method growth is here caused by HPV infection, human papilloma virus infection. When this virus entered in your body, its initial HPV infection, especially during the first year after therapy. precancerous changes long before invasive cancer develop. HPV virus into the sites. They will expand their cells to the peptides. Do not do the identification of that. After 8 years, it moves towards the cervical cancer. Okay? Now, human papilloma virus, long known to cause worms. These all are the worms. Very small. If somehow seen in your reproductive tract that this type of small worms is being happening, you must have to go to the primary health center or something. Right? So long known to cause worms found in many cancer foods. Ultimately, the HPV will make the cervical cancer. Over 100 types identified. How many HPV? More than 100 types of HPV, human papillomavirus. Different papillomavirus. Sometimes our child do not understand that the human papillomavirus does in her body or in her reproductive tract because of they are not harmful at all. They all are not harmful. Some of them are very harmful. So every year or at least 2 years or in the adolescence period, you have to do it. Most over 100 types of identified most benign. That means not much harmful. Benign, that means not much harmful. They will not make the cervical cancer. But to 20 can cause cancer. So when HIV enter in your body, don't sleep. Don't sleep. So when enter this virus, it is necessary every year for our girls, those who are belongs to 15 to 24 about these days, you must have to do the, or at least 30 years, you must have to do the practice. Lastly, I will let you know that very common 20 million current cases in United States. These new cases annually. 80% of women have HPV by age 50. 80%. It is not my data. I got from different literature. 50% of college students, repeatedly I am telling you, 50% of the college students, they are being affected by the human papillomavirus. But I have earlier mentioned that all papillomavirus are not harmful. Okay. Common HPV types and their effects. These HPV virus, that is HPV 40, 42, 43, 44, these all are benign. They are not much harmful. These type of human papillomavirus are not harmful. Not harmful. not harmful, but especially HPV 6 and 80. I have done my work on considering the HPV 16 and 18. These are very harmful. If it is entered in your body, obviously you do not take any necessary step. To some extent, ultimately it will be the causal effect of cervical cancer. Right? It will be the causal effect of cervical cancer. Now, HPV and cervical cancer, especially human papillomavirus, recognized as the underlying cause of cervical cancer since 1996. National Institute of Health, that is USA, Consensus Conference on Cervical Cancer in 1996, they have declared that if you do not take any necessary steps to control expression of the human papillomavirus, that will create a global burden for our women who will move towards the cervical cancer. In that conference, they had declared it. And World Health Organization and Europe and Research Organization, Genital Infection and Neoplasia in 1996, in the same year, they had declared that obviously you should have to take necessary steps controlling this disease. Otherwise, it will be a global burden, which will not beyond our control. Now, global burden. 5 lakhs women diagnosed every year. 2,70,000 deaths per year. If it is seen that the cervical cancer already being occurred in your body, it is too much to tough to keep that people or women in a living position. 1 million, and because of 5 lakhs women diagnosed every year, that they have already been affected by the cervical cancer. Out of this 5 lakhs, 2,70,000 deaths every year. 1 million new cases of cervical cancer each year. 1 out of 4 women who died due to the cervical cancer in the world is and my country. Is and my country. You will not be excited. I'll show you later. In Nigeria. What is happening in Nigeria? Just we have to follow here that in Nigeria, second most common cancer and second leading cause of cancer death among women in Nigeria. Incidence, approximately 12,075 women are diagnosed with cervical cancer annually. 12,075 women are diagnosed with cervical cancer annually. And mortality, around 7,968, 10,000 women die from this disease every year. Every year, Nigeria, 7,000 to 8,000 to 10,000 women die from this disease every year, with many causes of diagnosed death. And because of it has been diagnosed later. That's why it was in acute situation, the female candidate maybe. Now risk factor, over 60 billion age and older are at risk. Almost all are associated with the human papilloma virus. So this is the situation in Nigeria about it, whether it is a global burden or not. Now, this cervical cancer, we can prevent screening for precancerous changes. I have shown you the reproductive tract that when human papilloma virus entered in the position, that is the lower end position of the uterus, if it is being detected, and if you take necessary steps for prevention, obviously that will be cured. So screening is the most important, that is the practice for precancerous changes and treatment if problems found. Vaccination against HPV, that is prevention. When it is to be detected that HPV entered in your body, you just have to give, they will give you the vaccination. So it will not move towards the active cancer. And lastly, chemotherapy, radiotherapy, immunotherapy, all these. When patient is to be declared as that female candidate being affected very seriously, then you must have to treat that patient through chemotherapy, radiotherapy, and immunotherapy. We have done our work, just I am a little bit mentioning here. We have done our work in mathematical modeling. We have given chemotherapy and immunotherapy. That I will show you later. Not just a little bit knowledge of the cancer immunotherapy and updates, a paradigm shift in clinical oncology. General principle, the immune system recognizes and reacts against cancer. When earlier I have mentioned that when a foreign body particle enters in your body, our immune system is to be activated, and our immune system will try, especially the cytotoxic T lymphocytes I have mentioned earlier, cytotoxic T lymphocytes, and the antigen presenting cell that is called dendritic cell, CD8, who will give the signal to the cytotoxic T lymphocyte that the foreign body particle has entered in your body, so that these two are very much will be active. They will try to kill that foreign body particle. Human papilloma virus is a foreign body particle. So, immune system recognizes and reacts against cancer. The immune response against tumor is often dominated by regulation or tolerance. Evasion of host immunity is one of the hallmarks of the cancer. Some immune responses problem cancer both wild, short, depending. Defining the immune response against cancer will help in developing new immunotherapy. That means, after considering or forming the cervical cancer, immune systems will react as usual. There is another drug which I mentioned later, that immunotherapy. In a mild position, it will use that immunotherapy. In the precancerous state, then obviously you will get the epithelial. Now this is the T-cell response to cervical cancer. I am not going to this biological part much more intensively. But I must have to tell about this, the rejuvenating the immune system with PD-1 and PD-L1 blocker pathway, but PD-1 and PD-L1 blocker. That means when our latest drug, PD-1 blocker, PD-L1 blocker, when these virus to some extent That is, the virus growth is very high in the uterus. Some extent it will move towards the cervical cancer. During that period, if you keep the PD-1 blocker, PD-L1 blocker, what it will done? It will create a barrier. That virus will not impact the lower portion of that reproductive. So it will not happen, that cancer. Right? So we use after screening that it is the female reproductive tract is being nearer the precancerous stage. Just we have to use the PD-1 and PD-L1 block. Okay? Why did the T-cell need PD-1 blockade to attack the tumor? The anti-tumor immune response is a year-long struggle. The T-cell had tried, failed, and become exhausted. Why I am telling this? I am telling this in the flavor of the same thing which I have mentioned earlier, the basic philosophy, that when the foreign body particle entered in your body, always your immune system will be activated. Then dendritic cell, that is antigen presenting cell, will signal in everywhere. And these dendritic cells are very much associated with T cell, especially cytotoxic T lymphocyte. And that cytotoxic T lymphocyte basically try to kill the particle. He try and try. Is to be against the precancerous or against the infection of human papillomavirus. Thus, the anti-tumor immune response is a year-long struggle. When the human papillomavirus entered in your body, you will not be able to understand that I have been affected by the human papillomavirus and I have been being affected by the cervical cancer. After 1 year or 2 years, if you do not take necessary steps, then you will see. And that time, it is your out of control. And during this period, always cytotoxic T lymphocytes fight against the human papillomavirus as well as the cancerous cell. That's why I am struggling for my existence, but I do not get any money from anywhere. I am giving most of my things to the society or my workplace, but my workplace is not giving much money. So what will be my mentality? I'll be exhausted. In the same manner, T-cell fight, year-long fight, and the last day he is exhausted. And then cancer cell is to be closed. Now this is the PD-1 blocker. Cancer immunotherapy is different form of chemotherapy. I am not going to elaborate discussion of this. But another, this is the clone. And the latest work which has been published last year, I have not considered the clone consideration, but I have considered the stem cell. I have given a little bit flavor of stem cell in my previous class. This clone is mean uninfected or very healthy cell has been taken from your body, and the same cell can be given out in many. Suppose the cytotoxic T lymphocyte who is fighting against the HPV, just you have to take that cytotoxic T lymphocyte and mechanically you have to prepare cytotary in a massive level. Put that is the column. That cell you just have to put in your body. So much more cytotoxic T you have penetrated from the outside. That will kill the human papilloma virus as well as the cancer cell. Okay. This is the T cell white flags that can kill cancer cell. More is better T cell clonal extension. That is, clonal extension means if you create a T cell, if you are able to generate more and more T cell, then obviously that cell, if you enter in your body, but somehow it is to be entered in your body, then obviously that cell will react against HPV as well as cancer cell. The people may be against the harmful condition. This is the clonal extension, just you have to forget about this. And the future combination therapy. Future combination therapy, actually in my work, which I did, I have given two therapies in my mathematical model, where I experimental results from different peer-reviewed journals that I have to give some suggestions. That is immunotherapy, and the part is chemotherapy. That I have been given in my work, and this is my first work, which it was published in 2016, that how can HIV, HPV-induced cervical cancer can be controlled by a combination of drug therapy and mathematical strategy. It was published by my research scholars, along with one of my research collaborators, that how this was our first mathematical model, and you will not get much more mathematical model on HPV or cervical cancer. So what we did here we have considered SICVERA. What does it mean, S? S is the susceptible cell, which belongs to the epithelial cell, that is the upper portion of the cervix. Cervix, I have already shown you which portion is cervix. That portion cell we have considered as A. Can you remember the cervix portion? Just you have to remember the reproductive tract. Cervix portion is this. That cell we have considered. Susceptible cell is not infected. Now, the human papilloma virus will enter. Anyhow, that portion will be infected. We have considered that portion of cell is basically the susceptible cell, and I is the infected cell, and C is the cancerous cell, and B is the human papilloma virus. And another two is two drugs. On therapy, now the question is, all these biological issues, how you will formulate the mathematical model. In my discussion, what I have presented you, the cervical portion I have mentioned, considering during that portion of cells is susceptible cells. I have considered that HPV, that has been considered here as V. Consider these HPV will infect the epithelial portion of the leaves, which is portion of the uterus. That is infected cell, which is being infected by the human papillomavirus, and ultimately from that cervix to the uterus, to some extent it will create the cancer cell if you do not take any necessary state. So that I have considered that cancerous cell C. Another two cells I have considered, that is dendritic cell and the T cell. Why? During my presentation, I have repeatedly told that when the particles entered in your body, your immune will be activated, and always you will try to kill that particle. So obviously you must have to consider the T cell. And why this cell? This cell I have considered, this cell means dendritic cells. This cell, he basically what we do? He basically do that we give the signal, and it is called the antigen presenting cell. We give the signals to T cell. You go. I am telling you repeatedly. You read it. You follow my article and try to make new mathematical model. So dendritic cell try to same kind of words. Then you go to kill the human papillomavirus. That's why we have considered that area. And another two in my model R and H, which is basically the immunotherapy and the chemotherapy. Now, the H does not go in finite. Why? Why it does not go infinitely? Why? And because when it is growing, to some extent, that is to be converted to the H. Am I clear? So it must take the form of this infection, right? That's why we have taken S into 1 minus S plus I by K, where K is the carrying capacity and R is the intrinsic growth rate of whom? Intrinsic growth rate of susceptible epithelial, or this epithelial cell, that is susceptible cell. And when these cells interact with the virus with a particular rate, suppose it is probability, then it will interact or accumulate with each other. This is called the disease transmission following the law of mass action. From the total portion of logistic fashion, it will be reactive. And because of this interactivity, virus will be interacted with the susceptible cell. So that portion is to be reactive. That's why we have given minus A is B. Now, just you have to follow here. That is, this I interact with the susceptible cell. To some extent, it will be. I have not considered the latent field because the mathematical model will be more two-dimensional. I have not considered. I wish to change the dynamic. If you can consider, you can do it. So this term, where it will move? It will move towards the infected class. That's why it is moved to the infected class lambda is B. That lambda is B. Here it is moved towards infected class. And another term, just you have to follow me. That is this infected class. In this class, already I have mentioned that infected class, infected cell, always try to produce new virus. Am I clear? I have mentioned in my earlier class that when a cell is totally or actively infected, it will produce new virus, And that new virus, where it will move? It will move towards the viral class, or the human papillomavirus compartment, so that we have given this plus alpha 1I. Okay, alpha on I. And just you have to follow here. This is beta D1I. D1 just you have to follow this. Virus class, it is beta D1I. D1I means the minus beta I and minus D1I. It has the special instruction just I am giving you. D1 is the natural mortality of the infected class. And beta I, this beta, amalgamation of this beta will move towards the virus class, and again all these to be accumulated with this term. And again this term, that means the infected cell again, it is created due to the cause of new virus, which I have mentioned just 3 minutes ago. It will create more virus. That more virus will again infect the susceptible class, so the infected class will be gone. And that portion has been added here. Now, just follow me. This beta is the population rate. This beta is the population rate from infected to cancerous cell. This beta is the population rate from infected to cancerous cells. And this rho is the killing rate of infected cells by the cytotoxic lymphocytes. Or beta is the population rate of cancer. Whether I have written it right? I did. Why I have written it right? Can you tell me? So, no, just have to tell me about beta. Beta I. Why beta? Yeah, minus beta. Yes, minus beta I. Okay, beta I, right? Yeah. Okay. So minus beta I, at least infected cell, it might move to good. Infected cell, ultimately in my lecture, I have repeatedly told you that infected cell moves as a cell. So, at the beta rate, from the infected cell, it will move towards cancer cell. So, when I have prepared the cancer cell population in terms of mathematics, so this beta I minus beta I move towards this portion. It is okay? And D I, from DI, the from infected cell, they will produce more and more virions. Amalgamated. Here, eta G what DI. Eta means the number of free virions produced from the infected cell. Number of free virions produced from the infected cell. And this term minus rho IT, that means the T cell will kill infected susceptible infected cell, because I have earlier mentioned that T cell will try to kill the foreign body particle as well as the infected cell. Now, these cancer again will be grown, because cancer cell will not be in a stagnant position. When cancer cell is to be formed, he will make more and more cancerous cells. That's why it is fully affected by that region. And that cancerous cell is being produced more and more at the rate of A2. That's why we have written here class A2C. And every living organism, earlier I have mentioned, they have a natural mortality, so that it has the natural mortality into C. Now, dendritic cell. About the dendritic cell, in my previous class, I have told you that dendritic cell is an immune cell. T cell is an immune cell. It will produce from where? Bone marrow. Where it is to be mature? Thymus, right? Then it is to be directed different parts of the immune system, different position of the immune system via thymus, right? So it has a constant production. And that constant production we have considered here A. So, dendritic cell, why we have considered? And because of dendritic cell, it is the antigen present T cell. You will not. T cell. T cell. Virus has entered in your body. You do something. That's why we have considered the dendritic cell. So the philosophy of dendritic cell, how it is to be produced, from where it is to be produced, that should be noted. We know that it is a T cell, it is an immune cell, and it is to be produced from the bone marrow. It is matured in the thymus, then it is to be circulated different parts of the immune system. That's why it is S D T equal S minus M dt. Why we have written M dt? M means the rate of generator of CTL by DC. Rate of generator of CTL by DC. That means rate of generator of CTL by rate of DC by CTL. Sorry. Rate of generator of CTL by DC. So DC will inform me antigen capability. We borrow. You roll and try to kill the virus. That's why I have given this star is plus, so here not plus, minus MDT. And similarly, it has a mortality at the rate of D2, it can be given. Okay. Now, this equation. CDP. T means that is T cell, especially cytotoxic T lymphocyte. And this T cell we have considered R1 into IT into 1 minus T by K1 as because of R1. This R1 means the life growth rate of CTL cell. That the logistic growth rate of cytotoxic T lymphocytes and because of dendritic cell pushing him. So he will be enhanced and it will be grown. Intrinsic growth rate we are considering as R1. And it is to be happening due to the cause of here we have R1 IT into 1 minus T by K1. Why I have written IT? IT I have written because this first is trying to kill the infected cell. That's why we have given here. And another term we have given here that is first PMD. P means the rate of CTL proliferation induced by DC and M means the rate of generator of CTL by DC. So in that case what we did in the same pattern we have did it and it has the model that is it is D5P. Now we have considered two graphs. One is R and A represented the rate of decay of the dose. Rate of decay of the dose that means chemotherapy and we have considered the immunotherapy. Now this gamma R, what does it mean? The rate of clearance of the chemotherapy when this drug is to be imposed in this system, so it is to be declined. And that's why we have given minor gamma as a delta, that means the rate of clearance of the cytotoxic T lymphocytes activated drug when it will be imposed to the system, it is to be declined as because of it is acting to the system. And that's why we have given revoked as minor. So here, why it will react? It will react in this system as because of our target after consideration that the cancer has been formed. So we are giving the drug, the immunotherapy as well as the chemotherapy. So that impact has been reflected here and the cancerous cell is to be declined at a rate low, considering the chemotherapy, at a rate gamma, considering the immunotherapy. So this is the mathematical model. Now, I'll ask you whether you have questions to any question of this mathematical model. Just I would like to ask any question about this mathematical model. So this is our mathematical model. I have tried my level best to understand all of you. And what we did, we have got so many analytical results, so many numerical results. I am not going to this part. So many analytical results. We just will give you some flavor. What we received, that is our results suggest that strategy using double drug by impulsive approach can play a critical role in the dynamic evolution of cervical cancer. Mathematically, it is possible to bring the cervical cancer to a certain low level in the window of just 100 days. So we have achieved the result. You just have to use all these things, the chemotherapy and the immunotherapy, you will get better result within 100 days. And lastly, our what can help the biomedical field and enhancing the cytotoxic T lymphocytes and dendritic cell population, either by any drug or processes to a certain level to cure cervical cancer more quickly. So these two things was detected by calculation as well as analytical as well as numerical calculation. Our next work, it has been published, that is the role of HPV-Cancer with several response functions. I think no holding time 1, 2, 3, Professor Gage has already declared and mentioned, and that term we have considered in our mathematical model, whether we can get any dynamical changes considering this susceptible infected cell and the papilloma virus, and so that we have calculated a simple mathematical papillomavirus and cancer cell, and ultimately we have considered the holding type 2 response function here, and we have this. Our results suggest that the strategy using double optimal control play a critical role in the dynamic evolution of the cervical cancer. Mathematically, it is possible to bring cervical cancer to a certain low level in a window of just 200, which we have seen in our analytical as well as numerical result. Our work is biomedical enhancing tumor suppressor power of antibody process P53 and PRD either by any drugs or these two drugs actually we had considered during the process of calculation or other processes to cure cervical cancer more quickly. Now, another work which we had published in nonlinear studies, it was not a very big work, but we did therapeutic control of HPV-associated cervical cancer cell and possible extinction. Here we have considered the deterministic model as well as we have considered the stochastic model, and analytically we have seen it is possible to read the human papillomavirus in extincted level. That we have done in this mathematical model. Here only we have considered susceptible cells, infected cells, cancerous cells, T cytotoxic T lymphocyte, and human papillomavirus. We have given graph. This blocker we have given PD-L1 blocker and another blocker IL-2. I have mentioned in my earlier slide that interlinking to this medicine we had used in our mathematical model, and in a continuous phase we have given this with a optimal control approach. We have introduced this and we get some result. I am not going to this, and we are making this continuous deterministic system to a stochastic system, and ultimately we receive the result for extinction of human papillomavirus. That is why we can read our fundamental result of our mathematical model that we have proposed, both the deterministic and stochastic mathematical model that allows to simulate treatment scenarios and apply it to the specific case of immunotherapy of cervical cancer. And it is obvious the strength of our approach is to model mutual interactions and phenotypic state conditions of cancer cells and immune cells in a heterogeneous and dynamic micro environment in the context of therapeutic perturbations, increasing the death rate of the cancer cell and infected cell by combination of immune therapies can lead the cancer cells to become extinct, which is from the considering the stochastic model, and given the recent success of immune checkpoint inhibitors in cancer, it will be of our clinical interest to investigate the phenotypic evolution of the immune cells in the front of cervical cancer case. And lastly, we can say that our result suggests that stochastic events play under anticipated critical role in the dynamic evolution of the cervical cancer and the emergence of optimal therapy that requires further experimental and clinical investigation. And lastly, last year we have published this work, a mathematical this in cancer iPSC, which we have considered here in category, said that we have as a therapeutic approach, and here the stem cells which we have used in our mathematical model. Before going to the mathematical model formulation, I must have to say about the important pluripotent stem cell. What is the important pluripotent stem cell? It basically induced pluripotent stem cell, that is iPSC. It is abbreviated. We can say it is iPSC, are a type of cells that are generated by reprogramming adult cells, typically skin cells or blood cells, into a pluripotent stem. Pluripotent stem cells have the unique ability to differentiate into various cell types of the cells in the heart, brain, liver, and many others. Basically, this is a stem cell, and that stem cell can be derived considering any cell from your body. If you experimentally do it, you can derive this pluripotent stem cell from anywhere. That we have used in our mathematical model to control the cervical cancer. And the iPSC can generate it from a patient's own cell. The patient's own cells, he can generate this pluripotent stem cell, that is iPSC, which makes them patient-specific or autologous. Patient-specific means, suppose the human papilloma virus or cervical cancer patient, you just have to take the cell from that patient. You can produce the stem cell from that patient, or you are taking that pluripotent stem cell, you can generate it outside of the laboratory, that pluripotent stem cell, and you can inject that cell to control the cervical cancer of that particular patient, and which makes them patient-specific or autologous. The characteristic reduces the risk of immune rejection when these cells are used for regenerative therapy. And lastly, iPSC can be engineered to express specific tumor-associated antigens or differentiation, and these iPSC type cells can serve as a source for generating immune cells such as T-cells that target cancer cells, and this personalized immunotherapy approach holds promise for treating various kinds of cancer. Now, worldwide, the scientists are doing, or experimentalists is doing, considering the stem cell therapeutic approach. I have just given a little bit flavor, introducing the iPSC. It is a one kind of stem cell, induced pluripotent stem cells that I have introduced. It is not used still in the market. So what happens is still the experimentalists and the clinicians are doing this, and that results I have given in this mathematical model, considering their idea. So we have formulated this mathematical model. Here the same things we have considered the dendritic proportion. Only we have considered here the iPSC, that is stimulate pluripotent stem cells, that we have considered, and that model, that compartment we have added here, and we have get the result. We have received the result. We have applied the optimal control sequence with strategy T1, T2, T3, and this is T3 is the best strategy and efficient efficacy of the strategy. This and another is just I am telling this story about you about this what for formulating this mathematical model. We have given two drugs. One is immunotherapy, another is iPSC, right? And just we have checked numerically as well as analytically that whether if you do not give media, whether you can get or how type of results you can get. Later, this is T1. You are not giving any data. Later, we have used only iPSC. We are not giving the immunotherapy to the patient. We have checked. Again, we have given only immunotherapy, not iPSC. That we have checked. Again, we have given the two drugs combined. That is iPSC as well as the immunotherapy. We have received the best result. We have received the best result. That has been mentioned here. And Here, after 500 days, the treatment optimal control strategy T3, we achieve the maximum reduction in both the infected cell count and the cancer cell. That is infected with these two drugs. In strategy 3, when we have applied this, we have seen mathematically that HPV declination is rather than much more rather than the other model, as well as the cancer cell declination is much more rather than the other thing we have also been. That's why I must have to finish in this position. Before that, this book has been published in 2016 on HIV by speaker. You can follow it. Furthermore, I give a message to the audience that is, together we can fight cancer in a more blood-oriented approach with a mathematical and statistical science. And thank you very much.